So in this passage, we're going to go over how to effectively break down a biochem passage on the MCAT. This is the third section of the MCAT, and it will have many passages and passage-based questions. Some of these passages can be very information dense, so it is of utmost importance that you know how to break them down because there's also a time crunch and how to look at the passage and know when to balance looking at your content that you have been reviewing. So in this video, I'll show you how to break down the passage and answer the questions based on your previous knowledge and based on the information given in the passage, and how to interpret some of the discrete languages that the AAMC want you to break down. So here we have our passage, and we're going to do a quick scan of it. We'll highlight some keywords, and we'll answer questions with the keywords in mind. Many times I just read the first paragraph and look at just scan some of the other ones, and then move on to the questions. In the questions, I have this gridded box, which is where I'll write anything that I would typically write on the scratch paper that they give you on the actual exam. And in the passage, I'll write out any of the mental thoughts that I would have while reading the passage. So we'll get started. We're told that a 35-year-old presented with symptoms of fatty stools. So that will immediately tell you that fats are not being absorbed, right? So there's something wrong in either they're not being able to break down properly or they're not being absorbed, which happens in the small intestine. High in calories and carbohydrates. Duodenal biopsy revealed intact mucosal villi, so there's nothing wrong with the small intestine. Liver enzyme showed elevated levels of alanine transferase and aspartate. So we're told that our proteins are being broken down because our fats can't be absorbed, and our glucose levels, which we'll see down here, standard value, patient results. Standard value is about 95 or less than, and they're still very elevated. So that will tell you the hormones dealing with glucose are insulin and glucagon. If glucose levels are continuing to stay high, we know that there's something wrong with insulin because glucagon is when glucose is gone. So we'll have insulin in our mind as well. We'll continue on. Liver biopsy showed accumulation of fat droplets and hepatocytes and reduced staining for insulin. So that just shows insulin is not working or not being produced. One of those, right? Reverse transcriptase PCR analysis of the tissue showed presence of this virus. We know it's positive sense, single-stranded, and this point is good enough for me to move on to my questions. Now, if a question asks me specifics of this virus that we just read about, I will come back to the passage, but for right now, we can move on to the questions. So the first question is asking which hormone levels may be reduced in the individual described in the passage. The passage clearly told us that the hormone insulin, right here, reduced staining for insulin, but even if you didn't read this part, you can see this table and you can tell that since glucose levels continue to stay high, that something has to be wrong with insulin. So we'll pick insulin and move on. The best way to also look at this is you can look at the other choices and do a process of elimination. Glucagon is when glucose is gone, right? So the passage doesn't say anything about glucagon. It talks about how there is reduced insulin. So we can eliminate that. Aldosterone is when dealing with blood pressure. So we're not talking about that. We're talking about fats and everything. So we'll get rid of that. Epinephrine is in our sympathetic nervous system, and also it deals with glycolysis. No, sorry, glycogen. Um, so we'll not even look at that right now. So we know it's insulin. Moving on, according to the passage, these terms are super important. When it says according to the passage, we need to look at the passage because the passage will tell us what we need to know. The virus that we read about most likely disrupts disrupts the function of which structure in hepatocytes. So we have some options down here, and we'll go back in the passage and we'll look at the key words that will tell us which answer it is. So we know that it's a RNA virus, non-enveloped, encodes single polyprotein from a long genome. Doesn't tell us anything about a organelle. Continuing on, via receptor-mediated endocytosis, so maybe something with a plasma membrane, co-opts the host translation machinery. Translation is dealing with ribosomes. Translation occurs in the rough ER. So we know that this is ribosomes or rough ER, which is where ribosomes are. Continuing on, immunostaining revealed low expression of GLUT2 transporters, and reduced secretion of insulin that we've already told from pancreatic beta cells. Something's wrong with pancreas. Then down here, researchers hypothesize replication triggers ER stress and unfolded protein response. So that again confirms the rough ER part, because in the rough ER is where proteins are translated and they are folded correctly. So it shows that the replication causes, this is ER, which is endoplasmic reticulum, unfolded protein response, right? So we know that the answer is rough ER. It could be very tempting to pick smooth endoplasmic reticulum because they are talking about fats. It could be tempting to pick lysosomes because they talked about, endocytosis, and lysosomes are kind of like that. But the passage clearly states that there is endoplasmic reticulum stress and there's unfolded protein response. And there is a there's a host translation machinery hijacking of some sort. So we know it's the rough ER, and we'll pick that and move on. Okay, moving on to question 3. It's asking which structures did researchers observe in the biopsy of the liver? So this question is mainly did you read the passage? Did you read the passage, and did you catch what the researchers are finding out and what they are wanting you to get from the results? So did you read the passage? So in this one, we will go through our options, and it will be very helpful at this time in your scratch paper if we write that there is fatty liver, fats in liver. Then we know decreased insulin, and I would write it just like this on the actual MCAT to save me time, and I will know exactly what I'm talking about. There's decreased insulin staining, there is fat in liver, there is decreased GLUT2, and then there's increased blood glucose levels. And that was a breakdown from our study. Also, we'll just make a quick note, there's the HVX virus, positive ssRNA, and maybe that will be of help later, maybe it won't, we don't know, okay? So, back to our question, which structures did they observe? We know from our breakdown and from our reading of the passage that we observed C lipid droplets and reduce GLUT2 expression. Sorry, let me do this in green to keep the same color scheme. And we can also just confirm, like going back into the passage, we can just confirm right here. The immunostaining revealed low expression of GLUT2 transporters and reduced secretion of insulin, but at the top it told us that there is fat droplets observed in the hepatocytes. So we know that's true. We'll pick that and move on. It is, again, very enticing to see the other answers and want to pick that. Cholesterol deals with fats. Bios also deals with breakdown of fats. There's not breakdown of fats, so you would want to think crystals of cholesterol. That is not said anywhere, and that doesn't make sense either. Clusters of neutrophils and apoptotic bodies, that could also deal with the mild inflammation that they briefly said, or the immunostaining, but nowhere did the researchers observe that in the biopsy of the liver. Fibrosis and thickened hepatic sinusoids are also not talked about. So we will not pay attention to that and we'll move on. What type of enzymes did researchers most likely suggest the individuals be treated with? Again, we will add this to our breakdown of the passage. There is ER stress and increase in unfolded proteins. It says it right here. Researchers hypothesize that replication triggers ER stress and unfolded protein response. If proteins are being unfolded, and we can continue reading too, just in case we're missing something, ultimately interfering with hormone synthesis and secretion. They propose treatment with agents that reduce ER stress and restore protein folding capacity. Protein folding, this triggers the word chaperones because that is what helps in protein folding. So down here, we'll look for what matches our breakdown, which is C, chaperones that assist in protein folding. Protease that cleave polyproteins, the researchers did not recommend that. Kinases involved in insulin signaling. It clearly told us, this could be enticing, but it clearly told us that there is decreased insulin, and we even wrote that over here. There's decreased insulin, so a kinase involved in insulin signaling will not help them at all. The protease cleaving polyproteins won't help them either because they're all being made. They're just not being folded properly. So we'll take that away. And transfers that detoxify bilirubin, they did not even observe that in there. So we'll take that away as well. Then we'll come to this one. What is the approximately molecular weight of the polyprotein translated by the virus? When I come across questions like this, it's just a scan for numbers. Also, I want to make a note for question 4 is this is a background plus passage comprehension type of question. So many questions will need you to understand the passage and also go beyond and know your content. So watch out for those. And here we'll go back up and look at where we see numbers. Here we see the 7.5 kilobases. So we'll do that breakdown. 7.5 kilobases will translate to that divided by 3 to 2.5 kilo amino acids. So just 2.5 times 10 to the 3 amino acids. And that divided by 100 Daltons, it's about 200 something. And the minute I reach the first number or even like the second or something, and I see these vastly different numbers and the one that is closest to mine, see, I pick it and I move on, because if you know your calculation is right and if you're starting to get the right exponent or the right number, you can just pick it and move on. But it's best to calculate it all the way through if you're completely unsure. And then, which process is most likely observed in the HVX infected cells? Through our breakdown, we already know that there's something dealing with unfolded proteins. So we can pick B and move on. We also know rough ER is affected, so A is dealing with mitochondria. This is not correct because glycogenolysis could be a very valid thing because we don't have many glucose around, so we want to think like, oh, glycogen is breaking down, but that's not what's happening because the passage clearly says that the rough ER and the protein translation is what's being affected. Increasing DNA replication, that's not even talked about, usually is in cancer. This is not what we're dealing with right now. Which sequence of events does the virus most likely use to replicate in host cells? So this is where our, again, breakdown comes in hand because in our breakdown we wrote it's a positive single-stranded RNA. So positive single-stranded RNA will just pick the option that matches the replication of positive single-stranded RNA. Nuclear import transcription polyprotein synthesis, that does not sound correct because we know that positive RNA does not need to be replicated into another strand. Proteins can be directly translated from that using RNA-dependent RNA polymerase. And all we need after is replication of that RNA. So we'll pick direct translation, polyprotein cleavage, and RNA replication. So host RNA polymerase cDNA synthesis, this is typically of retroviruses, and that is not what we're told right now. We're told that it's a positive sense RNA. Transcription deals with DNA, and then we also have reverse transcription and integration. We don't need any of that. We know it's direct translation, and then the RNA replication. So we'll pick that and move on. And that's kind of just how you break down a passage, and you're using some of your background knowledge. We have written some of our stuff in our scratch paper, and it just gets pretty easy after you're answering one question, you're breaking down the passage and you're continuing, you get a good idea of what the passage is telling you, and using your background knowledge, you can easily answer any of the questions that they give you. That is all. You can send me an email here for any other questions. Thank you.